Hello friends, today we are going to study about composition, structure and function of carbohydrates, part 5. In this video, we'll learn about the folding of homopolysaccharide. First, let us understand how folding occurs. If we talk about the primary structure of any biomolecule, whether it is a carbohydrate or protein or lipids or nucleic acid, whether it is any biomolecule, the primary structure is always stabilized by covalent bond. If we talk about carbohydrate, then one sugar molecule is linked with other sugar molecule. For example, disaccharide. If it is a structure of maltose, then one glucose is linked with another glucose. If it is a structure of lactose, then one galactose is linked with glucose. Correct. If we talk about starch, then amylose and amylopectin, which consists of glucose units. Clear? So, when we are talking about carbohydrates, if it is a disaccharide, then one sugar molecule is linked with another sugar molecule. If it is a polysaccharide, then like this, we'll get a chain of sugar molecules linked with one another. So what this bond is? This is a glycosidic bond, which is a covalent bond. When we will study proteins, then we will come to know in case of protein, the basic unit of protein is amino acid. So one amino acid is linked with another amino acid and the bond is peptide bond. If it is a nucleic acid, then one nucleotide is attached with another nucleotide. In that case, this bond is a phosphodiester bond. If it is a lipid, then this bond will be ester bond, and all these bonds are covalent in nature. So the primary structure of any biomolecules, be it be a carbohydrate, be it be a protein, nucleic acid, lipids or fatty acids, everywhere we will see covalent bond. When this primary structure gets folded in a 3D form, during folding, the main stabilizing interactions that play important role are the weak interactions. Now already we studied about the bonds, weak interacting or stabilizing bonds. What bonds we studied? Hydrogen bond, hydrophobic bond, electrostatic interaction and van der Waals forces. So these are the forces that stabilizes the secondary structure or the 3D structure. Clear? The primary structure is always stabilized by a covalent bond. The foldings, the secondary structure, tertiary structure, or we can say the 3D structures, these structures are stabilized by weak interactions. Though they are weak interactions, but they are present in multiple numbers, hence they contribute to the stability of a biomolecule. Clear? So the subunits or the primary structure is stabilized by covalent bond and the 3D structures are stabilized by weak interactions, which include hydrogen bond, hydrophobic bond, van der Waals interaction and electrostatic interaction. In case of carbohydrate, the most abundant groups or set of molecules found is OH, hydroxyl group. In case of carbohydrate, OH is found in multiple numbers. Hence, which bond will contribute more among all the weak interactions? It is hydrogen bond. Okay, because in carbohydrate, OH group is found in maximum number, hence the hydrogen bond compared to other weak interactions contribute more for its stability. Now, this is the structure of a cellulose, where one glucose is bonded with another glucose and this linkage is beta-1,4-glycosidic. Now, in nature, the glucose molecules which are present in cellulose, they are present in particular rotations. They are not like a linear molecule. They rotate, they turn or they twist, correct? Creating some angles. So that what happens because of that turning or twisting? Because of that, the hydrogen bonds start forming. See this? The one glucose molecule is rotated 180 degree with respect to the next glucose molecule. Because of which the OH in these glucose molecules get arranged in such a way that they get involved in hydrogen bond formation, as we can see here. So the cellulose in nature, not only cellulose, all other carbohydrates as well, they are present in particular rotation. They are not like a linear flat sheet. They generally present, they turn, they get twist, and they present in a particular angle in nature. Correct. So in case of cellulose, one glucose molecule is turned 180 degrees with respect to the next glucose molecule, due to which all the OH groups in a glucose are get arranged in such a way that they get involved in hydrogen bond formation within each other. Clear? Now, as because they are turned in some particular angle, for example, one glucose molecule will have multiple groups of atom. Mainly, it will have OH, H, CH2OH, CHO, or C double bond O. These are the groups found in the sugar molecules. Correct. Now, this is a simple hydrogen atom. This one is a bulkiest group of atoms. OH is also bulky, CHO is also bulky. Correct. So, because any sugar molecule contain these bulky groups, therefore, rotation is not allowed at all angles. Okay. They cannot rotate 360 degree. Theoretically, they can rotate from 0 degree to 360 degree. But practically, all the angles are not favorable. The rotation around all the angles are not favorable. So some angles are favorable and some are not. Because of this, some structures will be, or some conformations are favorable. Now the angles which are favorable for sugar molecules for their turning and twisting, these angles are labeled as Phi and Psi. This will tell us the favorable angles at which the rotation is possible. This you might have learned, or those who have studied about Ramachandran plot of protein, these Phi and Psi angles we generally study in the Ramachandran plot. Here also we will study when we will study about the protein. Okay, but you might have heard about the Ramachandran plot. So what that plot says about protein to us, that the allowed conformations. The plot tells us what are the allowed conformations, because all the angles are not possible. The turning and twistings around all the angles are not possible because of what we can say. If because of CO2 a particular angle is not favorable, what we can say this type of hindrance? These are steric hindrance. What are steric hindrance? The hindrance that are associated due to the bulkiness or the heaviness or the large size of a molecule or the space they occupy, because of which if any hindrance is present, that type of hindrance is known as steric hindrance. So due to steric hindrance, the rotation around all the angles are not favorable. Therefore, all the conformations are not favorable. In case of protein, we study Ramachandran plot, which tells us which conformations are favorable for the proteins, where the angle, at which angle the turning and twistings are possible. Here also, in case of carbohydrate, all the angles are not favorable for turning and twisting of the molecule. Few angles are favorable and few angles are not favorable because of steric hindrance. Because they contain bulky or bigger size of group of atoms, therefore it creates steric hindrance at particular angles. Therefore, certain conformations are much more stable than others. Next, come to the 3D structure for starch and glycogen. Now, when starch or glycogen, it is mainly consists of amylose and amylopectin. So when they are twisted and turned in a 3D form or folded in a 3D form, they take the conformation of a coiled helix stabilized by hydrogen bonds. Why? Because we already studied in sugars, OH groups are more, therefore they are engaged in hydrogen bond formation. If we talk about amylose, the angle is 60 degree. That means one glucose residue is turned with respective to the next glucose residue with an angle of 60 degree. When that turning occurs like this, they are getting turned. So when these turning occurs, per turn, how many residues we can find? That is 6 residues. Now, because of this turning and twisting and this folding, a core is formed. In this core, iodine can fit either in I3 minus or in I5 minus form, ionic form. So, in this core, iodine can fit, and hence this is used as a qualitative test for detection of amylose. Now come to the structure of cellulose. In case of cellulose, if we talk about its primary structure, it is what? Glucose unit linked with another glucose unit. So this is a chain type of structure. Now, one glucose unit turned 180 degree with respect to the next glucose unit. Clear? In a chain structure. The turning occur as it is shown in the figure B. Now, what happens is, like this, several chains are present, where one glucose unit is turned 180 degree with respect to the next glucose unit so that it can form hydrogen bond between the sugar units in single chain and also between the sugar units of next chain. Correct? So we can find hydrogen bond intermolecule also and intramolecule also. Correct? Between two sugar units present in one chain and between two sugar units present in different chains. So like this, it forms a sheet having extensive hydrogen bonding, both inter and intramolecular. Because of this, it forms a super molecule structure having high tensile strength, and the water content is low. Why? Because all the OH are involved in bond formation among itself. All OH are involved in bond formation between glucose molecules found in cellulose itself. So no OH is available to form bond with the surrounding water. Therefore, the water content for cellulose is low. Now, one question from my end for you all. You have to tell me why cellulose is insoluble in water. It's a very easy question. I hope every one of you can able to answer this question. Do let me know in the comment section. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.